Oparin thought that he could explain the origin of the first life using Darwinian principles. He envisioned simple chemicals combining and recombining to form larger molecules, and then these larger molecules organizing themselves with the help of chance variations and natural selection into the first primitive living cell.
Over the next 3 decades, many scientists worked to develop and refine these ideas as they pondered the questions both Oparin and Darwin had raised. How could life have evolved from simple chemicals? One man thought he knew.
The problem of biological origins has, for a very long time, I would say, has been a real deep interest to me just because of the scale of the problem, the importance of it. Where did we come from? Why are we here? All that kind of question. Probed from the point of view of natural science.
During the late 1960s and throughout the 70s and early 80s, Dean Kenyon was one of the leading chemical evolutionary theorists in the world. And like others in his field, he was trying to explain how life on Earth began through a purely natural process.
In 1969, Kenyon co-authored an important book on the origin of life.
Gary Steinman and myself thought that if we were to pull together in all of the lines of empirical evidence that had accumulated by the mid to late 60s in one continuous argument, we were very enthusiastic about the possibilities for explaining the origin of the main life-building elements.
Despite his optimism, Kenyon faced a significant problem. To explain how life began, he first had to account for the origin of the essential building blocks of every cell that has existed on Earth. Large, complex molecules called proteins.
Proteins have a wide range of function in the cell, everything from structural requirements in terms of scaffolding of the cell, the cytoskeleton, to enzymes, where they're actually processing molecules to harvest energy or to build components of the cell.
Proteins do pretty much all of the jobs inside of the cell, except for storing genetic information. That's left to the DNA, the RNA. But all the day-to-day jobs, cleaning up the cell, making energy, it's all proteins.
Kenyon knew that proteins would have been as important to the first life as they are to living cells today. He also recognized the complexity of their construction.
By the 1960s, scientists had determined that even simple cells are made of thousands of different types of proteins, and the function of these molecules derives from their highly complex three-dimensional shapes. The irregular shapes of some proteins allow them to catalyze or trigger chemical reactions because of the hand and glove fit that they have with other molecules in the cell, while other protein molecules form interlocking structural components.
The individual parts of a bacterial motor, like this ring structure, are each made of either a single protein molecule or an assembly of proteins fitted together into a specific shape. These proteins are, in turn, made of smaller chemical units called amino acids that are linked together in long chains.
There is a very great degree of intricacy of architecture down in the cell units in these protein-forming amino acids.
In nature, 20 different types of amino acids are used to construct protein chains. Biologists have compared them to the 26 letters of the English alphabet.
Alphabetic letters can be arranged in a huge number of possible combinations, and it's the sequential arrangement of the letters that determines whether you have meaningful words and sentences. If the letters are arranged correctly, you'll get meaningful text. But if they're not arranged correctly, you'll get gibberish. And the same principle applies for amino acids and proteins.
There are at least 30,000 distinct types of proteins, each made of a different combination of the same 20 amino acids. They are arranged, like letters, to form chains, often hundreds of units long. If the amino acids are sequenced correctly, then the chain will fold into a functioning protein.
Proteins are arranged with their amino acids in such a way that the amino acids collapse on each other into an architecture that is pre-programmed by the order of the amino acids. It folds into a certain structure, and that structure can do a certain function. So all proteins in the cell have a certain three-dimensional pattern that's based on the arrangement of amino acids in the chain.
This arrangement is critical, for if the amino acids are incorrectly sequenced, a useless chain forms, and instead of folding into a protein, it will be destroyed in the cell.
Proteins, like written languages or computer codes, have a high degree of specificity. The function of the whole depends upon the precise arrangement of the individual parts.
But what produces the precise sequencing of amino acids that gives rise to the specific shapes and functions of proteins? During the 1950s and 60s, discoveries about protein structure forced biologists to confront this mystery. Dean Kenyon believed he could solve it. In his book Biochemical Predestination, Kenyon and his co-author Gary Steinman proposed an intriguing theory. Kenyon wrote, "Life might have been biochemically predestined by the properties of attraction that exist between its chemical parts, particularly between amino acids in proteins."
At the time that biochemical predestination came out, I and my co-author were totally convinced that we had the scientific explanation for origins.
Kenyon proposed that the chemical properties of the amino acids caused them to be attracted to each other, forming the long chains that became the first proteins, the most important components in the living cell. And this meant that life was effectively inevitable, predestined by nothing more than chemistry.
Many scientists embraced Kenyon's ideas, and over the next 20 years, Biochemical Predestination became a best-selling text on the theory of chemical evolution. Yet 5 years after the book's publication, Kenyon quietly began to doubt the plausibility of his own theory.
It was during that whole period of time that my doubts about certain aspects of the evolutionary account became apparent. When coming into contact with a powerful counter-argument given to me by one of my students, and I could not refute that counter-argument.
Kenyon was challenged to explain how the first proteins could have been assembled without the help of genetic instructions. In living cells today, chains of amino acids are not formed directly by forces of attraction between their parts, the scenario Kenyon envisioned on the early Earth. Instead, another large molecule within the cell stores instructions for sequencing the amino acids in proteins. It is called DNA.
Initially, Kenyon believed that proteins could have formed directly from amino acids without any DNA assembly instructions. And that's why so many scientists were excited about his theory. But the more he and others learned about the properties of amino acids and proteins, the more he began to doubt that proteins could self-assemble without DNA.
In DNA, Kenyon encountered a molecule with a property he could not explain through natural processes. For locked securely within its double helix structure is a wealth of information in the form of precisely sequenced chemicals that scientists represent with the letters A, C, T, and G. In a written language, information is communicated by a precise arrangement of letters. In the same way, the instructions necessary to assemble amino acids into proteins are conveyed by the sequences of chemicals arranged along the spine of the DNA. This chemical code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe.
Like other scientists working on the origin of life, Kenyon realized he had two choices. Either he had to explain where these genetic assembly instructions came from, or he had to explain how proteins could have arisen directly from amino acids without DNA in the primordial oceans. And in the end, he realized he could do neither.
It's an enormous problem how you could get together in one tiny submicroscopic volume of the primitive ocean all of the hundreds